Believe it or not, back before and up into the 1600s, there was this idea known as spontaneous generation to explain where life comes from. And the idea is that life will spontaneously generate from non-living matter in a very short period of time. So it's important to understand that. So, for example, like we see in the lower left here, that maggots will spontaneously generate from meat, that fish will spontaneously generate from the mud in the bottom of a pond, that microorganisms will spontaneously generate in spoiling broth and like grape juice to ferment it into wine and so forth. So that's the idea. So Reddy was a scientist who designed an experiment to test the idea that maggots spontaneously generate from meat. And he thought that that's not where maggots came from, because he observed flies landing on meat that was left out. And he thought that even though he couldn't see the fly eggs are so small that you can't see them. You need a microscope to see them. But he thought that even though he couldn't see the eggs, that the flies were laying their eggs on the meat, and that the eggs then would hatch into the maggots, which are fly larvae, and then those maggots would metamorphose into flies, which we now know is the life cycle of flies. So he hypothesized, so that was his observations. So then he hypothesized, and this would be the alternative hypothesis: if maggots hatch from fly eggs and we allow meat to age in a covered jar, then maggots will not spontaneously appear. Now the cover in this case would be something like a screen that would allow air to come in but not the flies, to keep the flies out. A null hypothesis for this experiment would be that there would be no difference even if you covered the jar. So if maggots do not hatch from fly eggs, in other words, they spontaneously generate, and we allow meat to age in a covered jar, then maggots will appear just as they do in an open jar. So in other words, no difference between the control group and the experimental group. That's what the null hypothesis predicts. Nothing will be different. So he set up this experiment with 2 jars, so everything's exactly the same. You know, if we assume that this is a good controlled experiment, everything's exactly the same. Same jars, same piece of meat, same amount of meat, everything. He tried to control everything. The only difference is that one jar is covered and the other jar is not. The one that is not covered would be the control group. The one that is covered would be the experimental group. And if you're trying to identify the groups, the experimental group is the one that you apply the independent variable to. And in this case, the independent variable is whether or not there's a screen. So you're applying a screen. The jar that you're applying the screen to is the experimental group. We can also assume that he did this at least multiple times, or he had multiple jars each time he carried out the experiment. Because, as I've told you before, one experiment proves nothing, and the more you repeat it, the better it will support the hypothesis or refute it. So the dependent variable is whether or not there are maggots, whether or not maggots appear. Because whether or not maggots appear depends on whether or not there's a screen. That's the relationship between the independent and the dependent variable. So he concluded that maggots come from eggs laid by flies, which is what he had inferred by observing the flies landing on the meat, and that they do not spontaneously generate. And we can assume that his experiment is valid, like I said, as long as he controlled all other variables, and he did. Not long after Reddy's experiment, a scientist named Needham said, okay, so Reddy showed that maggots don't spontaneously generate from meat, but I think that microorganisms will spontaneously generate from a broth or gravy to spoil the broth or gravy. So he set up an experiment where he had a flask filled with gravy, and he heated the flask, heated the gravy in the flask, and then sealed it off so that nothing could get in. And time passed, and he saw microorganisms appear in the gravy. So being skeptical and critical, we see now that this experiment was not valid. And it's because he only heated the gravy. He didn't boil the gravy. We know now that boiling is what kills the microorganisms in anything. That extreme heat will kill microorganisms. And he also only had one group. He only had an experimental group. He didn't have a control group. His results and conclusions contradict Reddy's, but his results and conclusions are not valid because he didn't conduct a good controlled scientific experiment. Spallanzani looked at Needham's experiment and said, no, that, he realized the same thing that we realize today, that experiment was not valid. So he set up an experiment with two groups, an experimental group, a control group, and he boiled the broth, or boiled the gravy. And his results show that microorganisms do not spontaneously generate. After boiling and sealing the flask, microorganisms did not appear, therefore refuting the spontaneous generation hypothesis, just like Reddy's experiment with maggots refuted spontaneous generation. So even so, we've got these two well carried out experiments to show that spontaneous generation doesn't happen, but there were still scientists in the scientific community that were being critical and skeptical, and they said, well, maybe there's this vital force in the air of the flask that gets destroyed by boiling, and that vital force, once it's destroyed, cannot cause spontaneous generation to happen. So we needed one more experiment to show that there is no such thing as a vital force in the air that allows for spontaneous generation. And that was carried out by a scientist named Pasteur. So along comes Louis Pasteur, and he feels, he hypothesizes that there is no vital force in the air, and that the microorganisms actually come from particles in the air. And we know today that they do, that microorganisms like bacteria produce spores, and those spores then fall into a broth or a gravy or the grape juice that ferments in the wine or whatever, and that's where the microorganisms that appear come from. They come from the spores. Same with fungi, they produce spores. But those are invisible, and at this time, it was basically around the time that microscopes were first being invented, so they couldn't see those things. So Louis Pasteur designed an experiment to show two things: that there was no vital force, and that microorganisms come from particles in the air. And the way he did that was to invent a different kind of flask that had a curved neck. And he hypothesized, basically had two hypotheses. We're not dealing with a null hypothesis here, so these are both alternative hypotheses. So if there's no vital force allowing for spontaneous generation, and we boil the broth in an unsealed curved neck flask, so it's not sealed like Needham and Spallanzani used, but it does have this curved neck, then microorganisms will not spontaneously appear in the broth. And then another hypothesis is that if microorganisms come from particles in the air, and we boil the broth in an unsealed curved neck flask, then the microorganisms will not spontaneously appear in the broth. So the idea here is, with this curved neck flask, that the particles in the air, the spores, the microorganism spores, can't make it past this curve in the neck of the flask. They can't go all the way past this curve and all the way up this tube and all the way to get to the broth. So he set up the experiment just like Needham's and Spallanzani's. The only difference really is this curved neck flask that he invented to try to solve this problem. So it's still open to the air. So if there's any kind of vital force in the air that causes spontaneous generation or allows spontaneous generation to take place, then the vital force can still be there. but what he did was block the ability for any particles of microorganisms to get to the broth. And he boiled both of them, so the control group was unboiled, the experimental group was boiled, just like in Needham and Spallin, or just like in Spallanzani's experiment. And that's the independent variable. The dependent variable is whether or not microorganisms appear in the broth. And microorganisms did appear in the broth after a period of time in the flask that was not boiled, because the microorganisms weren't killed in the first place. In the flask that was boiled, the microorganisms did not appear until the curved neck of the flask was broken off. So this could sit for a year without spoiling. Even though it's open to the air, the particles, the spores from microorganisms are not able to get up the curved neck flask and into the gravy or the broth to spoil it, even for a long period of time, until you break off that curved neck. And then the microorganisms are able to fall into the broth and spoil it. So Pasteur's experiment is valid, and we revere him for putting really the final nail in the coffin of this whole idea of spontaneous generation. And his results contradict Needham's, but Needham's results are not valid, and they agree with Spallanzani's and Reddy's. So this was, again, the final experiment to show that there is no spontaneous generation, that life does not come from non-living matter in a very short period of time, spontaneously. So not all scientific questions or hypotheses can be supported or refuted by a controlled experiment or a scientific experiment. And an example of that is animal behavior, or questions like this one. And I chose this question to put on this slide because a girl last year did this experiment for her year-long research project. She went out and observed the population density of black squirrels at parks further and further away from Kent. So she went away as far away as 25 miles. So she measured the squirrel populations up to 25 miles out from Kent to compare how the population density changes as you go from Kent, where black squirrels were first introduced, supposedly, out further away from Kent. And that's something that you can simply measure or observe. It's nothing that you can do a controlled experiment where you have a control group and an experimental group. Likewise for animal behavior, and a lot of times now to find out what's going on with animals and how animals are behaving, we put trackers on them and put cameras on them. And these are actually older images where the cameras are really big. Now the cameras are much smaller that are being used for that. But those are examples of scientific questions that really can't be answered by experimentation. And, you know, like what we know about dinosaurs, all has to simply be inferred. We can't do experiments with dinosaurs. We could do experiments with reptiles, which are relatives, the living reptiles today, which are relatives of dinosaurs, but we can't go back in time and do any experiments with dinosaurs. So there are also ethical concerns with using living organisms, of course, because if you use organisms for your experimentation and you cause them discomfort or pain or even death, that has to be taken into consideration. And of course, there are organizations like PETA and so forth that are very concerned about that kind of use of animals in scientific experimentation, especially our close relatives like the primates. You know, this is a really pitiful image here of two monkeys that appear to be just petrified with some kind of apparatus on their head for an experiment. And the reason this picture is here is the cosmetic industry really came under fire a number of years ago because they were using, for example, these rabbits in what looks like something like a stockade, where they would shave a part of their fur and then put these cosmetic substances onto their skin and they're, you know, it basically amounted to torture of those poor rabbits to carry out their experimentation. So you can't use humans, you know, we can't use humans to test these things, so we have to use stand-in species, so to speak, what we call experimental models to model results that might be comparable to humans. So we need models that are closely related to humans. And if you look at the pie chart here, and the image here represents that we use rodents very heavily in experimental research, primarily mice. And the reason is that mice share about 85% of their genome, in other words, their genes, with humans. So mice and humans are about 85% identical in their genes. So if we find a particular result in mice, then we can assume to some extent that we would expect the same results with humans. And that's the whole idea of experimental models. Now we're going to shift gears a bit and start looking at biology very generally, and why biology is so important to you. Obviously, that's what this slide is about. Why should you care about biology? Well, it's the study of life, and you're a living thing, right? So it's the study of you. Biology is the study of you because you're a living thing, and it's a study of every other living thing. You should be concerned about biology because you interact with other living things. You're a living thing. There are many other living things, and there's a lot of interaction going on between you and other living things. Whether you know it or not, you may say, oh, I never go outside and I never see other animals or anything or come in contact with other animals, but you do come in contact with other living things because there are billions of bacteria that live on and in your body and other microbes that are interacting with you all the time. You eat living things, right? That's really important to you. Food. Agriculture is a subdivision of biology, where your food comes from. Earth is habitable to you due to living things. If it weren't for the fact, for example, that photosynthetic organisms produce oxygen as a byproduct of photosynthesis, you wouldn't be here. Modern medicine and biotechnology is helping to keep you alive and will help to keep you alive, especially as you get older and older. So those are all reasons that biology, you should take biology seriously. And I would say that the most potentially transformative biotechnology that's currently being developed is the ability to manipulate our own genome. In other words, we're taking control of our own evolution, as well as the genomes and evolution of other species. And that's a very powerful thing, extremely powerful thing. So we are a species that has evolved to the point where we have the capability of controlling our own evolution. That's an incredible thought. What we have here is a short list, actually, of sub-sciences that fall under the umbrella of biology. And I'll leave you to think of any that might not be on the list, but there are many that are not on this list. So if there's anything, the point here is if there's anything that you are interested in, whether it be art, or even music, there is a place for you, or math, or chemistry. So you may, really like chemistry when you take chemistry next year, but we need biochemists. So that's an example of how you could apply your talents to any one of these sub-sciences that fall under the umbrella of biology. So that's the point of the slide. It's a very humongous field. It's a humongous field and a big umbrella that encompasses a lot of subfields of study. So you are needed. So there's a place for you under the umbrella of biology. So what is a living thing? That's a tricky thing to define. But this is one list of many lists and many criteria that have been developed to try to explain what a living thing is. But you can put this phrase in front of everything on this list that I've generated, and this is a list that somewhat came from your textbook, but I've added to it. So life as we know it is composed of one or more cells. So there are one-celled organisms and there are multicellular organisms, or in other words, there are unicellular organisms and multicellular organisms. But one cell, the cell is the basic unit of life. So there is nothing that is considered alive that is less than at least one cell. Life as we know it has DNA. Every living thing has DNA and uses the same genetic code, which suggests something really extraordinary, and that is that all life on this planet is related to the very first life that ever appeared on this planet. Life appeared on this planet, and then all life that is now here arose from those first living things. Life as we know it reproduces. Whether it be asexual or sexual reproduction, living things reproduce. Otherwise, they would go extinct. Life as we know it grows and develops. Life as we know it obtains and uses energy to organize matter. That's what we call metabolism, and that's a whole unit of study. A lot of these areas here, we're going to be studying reproduction and growth and development and metabolism. So a lot of these characteristics that make a living thing are representing areas of study that we're going to be studying this year. Life as we know it maintains homeostasis. That's a big word, but it simply means stay the same. Homeo means the same, stasis means to stay, right? So stay the same. In other words, maintaining a stable internal environment, whether it's a one-celled organism or a multicellular organism. Living things need to maintain a stable internal environment in order to stay alive. Living things interact with their external environment. That's the whole field of ecology. Interaction between living things and other living things, and living things and even non-living things is ecology. But living things interact with their environment. Living things evolve, change over time, and that's necessary. Again, just like if living things didn't reproduce, they would go extinct. Living things evolve or they go extinct. Living things have had to evolve on this planet because the planet has changed over time in a big way. So living things have to change also in order to keep up with the changes of the earth. And finally, living things require water because the biochemistry of life takes place in aqueous solution. That means things dissolved in water. An aqueous solution is a solution where the solvent is water and there are solutes, substances dissolved in that water, and all of biochemistry takes place in water. So, that's why we're looking for evidence of water on Mars, for example, because if there is water on Mars, or evidence that there has been water on Mars, that suggests that there may be life on Mars, or maybe there used to be life on Mars. So the idea here is that all of these must be present to define life as we know it. So what is something that is kind of like a biological entity that is not considered living? Most biologists consider viruses to not be living. So I know this diagram, when you first look at it, might look like a cell and therefore maybe represent a living thing, but it's actually a virus. And viruses are considered non-living biological particles. Because they are obligate intracellular parasites. They can't do many of the things that are on this list by themselves. They have to use a living cell in order to perform these characteristics of life. So they're not composed of cells. A virus is not a cell. So they violate number one. They kind of violate number two. Some viruses don't have DNA. Some viruses only have RNA instead of DNA. And that's the example that we're looking at here. It's an RNA virus, not a DNA virus. Number three, viruses don't reproduce by themselves. They use living cells to replicate. So I don't even use the word reproduce for viruses. I use the word replicate. They replicate in living cells. Number four, then, grows and develops. Viruses don't grow and develop. They are assembled in living cells, and once they're assembled, they don't change. And then finally, number five, viruses don't obtain and use energy on their own. They hijack living cells and And the cells are using energy to organize matter. And they use, so the viruses are hijacking the cell to use the cell to organize themselves, to make themselves, to replicate themselves, to build themselves. So they really have no metabolism of their own. So that's why it says versus, right? Living things versus non-living things. And a virus is an example of a biological entity or biological particle, but it's not considered by most biologists to be a living thing itself.